Anyong Liam, this is Liam in for bear. When a cell runs out of oxygen, it fires an emergency program. But what happens when that emergency program runs permanently in a cell with plenty of oxygen? The answer is a broken off switch, and it explains clear cell kidney cancer. Can you explain it, Bear?
Clear cell kidney cancer behaves as if it is starving for oxygen, even in tumor regions with plenty of it. A biopsy of a clear cell renal tumor shows high GLUT1, high VEGF, a full hypoxic adaptation program running. Then you measure oxygen in that region. Partial pressure, adequate, normal. The cells act starved. The oxygen is there. The driver of the hypoxic program is HIF-1 alpha. It accumulates when oxygen falls and drives transcription of glucose transporters, glycolytic enzymes, VEGF. In a well-oxygenated cell, HIF-1 alpha should be essentially undetectable. Here is the mystery. The HIF-1 alpha gene is intact in clear cell kidney cancer. The oxygen sensors, the Prolyl hydroxylases, are intact. Oxygen is adequate, yet HIF-1 alpha is constitutively active. Why does a cell with normal oxygen sensing fire the hypoxic program anyway? The naive expectation: if the oxygen sensor is intact and oxygen is adequate, HIF-1 alpha should be destroyed on schedule. The sensor fires. The destruction pathway should complete. The cells should not be hypoxic. So where does the path break? Not at oxygen, not at the hydroxylase. The failure is one step downstream, the protein that reads the hydroxylation tag, VHL. In a normoxic cell, the Prolyl hydroxylases add a hydroxyl group to two proline residues on HIF-1 alpha. That tag is the signal. VHL is the reader. VHL binds the hydroxylated HIF-1 alpha, recruits a ubiquitin ligase, and commits the protein to proteasomal destruction. HIF-1 alpha has a half-life of roughly 5 minutes. Now it is made and immediately gone. Clear cell kidney cancer loses VHL through deletion, mutation, or methylation in nearly all cases. Without VHL, HIF-1 alpha is hydroxylated on schedule. The tag is there, but there is no reader. The destruction machinery is never recruited. HIF-1 alpha simply does not get destroyed. The result is constitutive HIF-1 alpha activity, not because the cell is hypoxic, but because the off switch is broken. The sensing step completed. The disposal step is missing. HIF-1 alpha floods the nucleus and runs the full hypoxic transcriptional program in a normoxic cell. Here is the comparison side by side. Left, a normal cell. Oxygen adequate, PhDs active, VHL present. HIF-1 alpha is hydroxylated, recognized, destroyed, functionally absent. Right, a VHL null clear cell RCC cell. Same oxygen, same PhDs working. VHL absent, HIF-1 alpha hydroxylated, and simply never destroyed, constitutively active. The cancer is not detecting low oxygen. The cancer has lost the protein that reads the signal and acts on it. The oxygen sensor reports correctly. The report goes nowhere. The consequence is the entire hypoxic adaptation program running at full throttle in a well-oxygenated tumor. GLUT1 upregulated, glucose floods in. Glycolytic enzymes upregulated, carbon routed to lactate and building blocks. VEGF secreted, new blood vessels recruited. The Warburg metabolism, the clear lipid-loaded cytoplasm, the high vascularity, all of it downstream of one missing protein. Here's the worked example. In a normal kidney cell, HIF-1 alpha is synthesized continuously. The Prolyl hydroxylases tag it in about 45 seconds. VHL binds in about 10 seconds. The proteasome destroys it. Net half-life, roughly 5 minutes. In a steady-state normoxic cell, HIF-1 alpha is essentially undetectable. In a VHL null clear cell RCC cell, same synthesis rate, same Prolyl hydroxylase activity. HIF-1 alpha is hydroxylated on schedule, but VHL is absent. The hydroxylated protein accumulates. After 4 hours, it is at roughly 40 times normal levels. GLUT1 expression rises. VEGF is secreted. Glycolytic enzymes upregulated. The oxygen sensing circuit completed its detection step. The disposal step is simply missing. VHL is the recognition protein that reads the hydroxylation tag and commits HIF-1 alpha to degradation. Lose VHL and HIF-1 alpha piles up constitutively regardless of oxygen. running the entire hypoxic adaptation program in a cell that has plenty of air. A broken off switch, not a false alarm.
VHL is not the sensor, it is the reader. PhDs tag HIF1 alpha, VHL converts that tag into a destruction order. Lose VHL and HIF1 alpha accumulates regardless of oxygen. One missing protein, whole hypoxic program permanently on. The trade-off that makes this devastating is the same one that makes normal oxygen sensing elegant, a single bottleneck, efficient until it is gone. Your turn, take this prompt into Claude and explore what happens when you stress test the VHL-HIF axis from a drug design angle.